Well, good morning, everyone. It's truly an honor to be here. On behalf of the other laureates, let me tell you that we are all deeply honored for the way we are being treated and recognized in an incredibly warm fashion. I'm going to tell you about the work that was really completed by hundreds of scientists working around the world, as you heard in the introductory video, an effort known as the Human Genome Project. I'm simply here to represent their collective efforts and their collective accomplishments. Growing out of a very young field of genomics, which was first actually named as a discipline in 1987 in a lead editorial of a new journal called Genomics, was this notion of we had the tools in hand for now being able to essentially read out the complete blueprint of organisms, and the complete blueprint is all the DNA of an organism, that is a genome. And the idea of comprehensively studying the genome of organisms really came to fruition in the late 1980s, and the field was born. What the honor that was given to the field of medicine and the group of scientists that were involved in this effort was the notion of putting together an international effort called the Human Genome Project, where scientists from around the world would come together in a very focused way to try to elucidate the complete sequence of all the letters in the human genome. Roughly 3 billion letters, or building blocks, are what make up the human genome. You heard about some of this in the opening video, but in essence, the project was remarkably successful and over a 13-year course essentially completed all of its goals, including determining the order of these 3 billion building blocks that make up the human genome. Rather than telling you the details of this effort, which was completed 15 years ago, I thought it was more important to set a broader context of what is happening now in genomics, with it basically having its first 3 decades of accomplishments to catalog. Now, the lead highlight in the field of genomics in its first 3 decades is the completion of the first sequence of a human genome completed by the Human Genome Project. But really, that was just the first of many highlights and just the first accomplishment of genomics, because what then has transpired in the last 15 years has been an amazing suite of applications of genomics that have touched many, many different areas, a subset of which are shown here. And in fact, some of this, for example, the use of genomics to study infectious agents, I am quite sure will be heavily emphasized in this conference. What I'm going to tell you about are some of the applications of genomics even beyond the 9 shown here, particularly with an emphasis of the use of genomics for studying human health, human disease, and eventually the practice of medicine. And in fact, what we have turned our attention to over the last 15 years has been basically thinking about how to use genomic information about each individual person to better think about how to treat them and take care of them medically. This has led to the notion of a new discipline of medicine, which I will refer to as genomic medicine, which is basically the idea of not treating patients generically, but rather recognizing that each of us has a slightly different genomic blueprint. And if we could capture information about each of our unique features in our genome, can we better tailor the way we take care of our patients? This is really viewed as a journey. It's a journey that began with the Human Genome Project. That was really our starting line. And we recognize that it'll take many years and many steps to fully realize genomic medicine. But as soon as the Genome Project was completed 15 years ago, it was very clear we could begin walking down this path towards eventually realizing genomic medicine, albeit recognizing that we won't know exactly what all the steps are along the way. We also recognize that, as with the Human Genome Project, where large numbers of scientists from different disciplines were needed to work together to accomplish a common goal, realizing genomic medicine will similarly take not one laboratory, not one country, but many laboratories, many countries, many different disciplines to accomplish this important goal. This is more like a marathon as opposed to a sprint, and many, many people, scientists, clinicians, and so forth will need to be running for a long time, shoulder to shoulder, heading towards the realization of genomic medicine. Well, with this as an aspirational journey, how do we progress from the Human Genome Project to the realization of genomic medicine, what have been some of the other highlights in the first 3 decades of genomics? Well, I would tell you that the next highlight, beyond having completed the Human Genome Project, relates to the fact that we didn't just want to sequence one human genome. We eventually want to sequence many, many people's genomes, and we want to do it as part of routine medicine. In order to do that, we needed to reduce the cost of sequencing DNA. And what I can tell you is over the last 15 years, we have seen stunning technical advances in methods and technologies for sequencing DNA, such that we have seen the cost of sequencing a human genome reduced by nearly 1 million-fold. Now, let me emphasize, the Genome Project was incredibly successful at its goal of sequencing a human genome, but it was an expensive endeavor doing it for the very first time. In fact, that first human genome sequence cost something like $1 billion, and yet $1 billion was not going to be a practical clinical test in the care of our patients. So we saw it as a goal to reduce the cost of sequencing a human genome to something like $1,000. And the last 15 years, through a series of major innovations, one after the other, we have seen continual decrease in the cost of sequencing DNA, such that now we are practically at the point that we can sequence a human genome for something like $1,000. Now, it's not just the cost of sequencing DNA, it's also the recognition that we need to be able to sequence DNA quickly as part of clinical care. That first human genome sequence accomplished by the Human Genome Project, which cost $1 billion, was also slow. It took us 6 to 8 years and hundreds of people working in big laboratories to be able to actually read out all the letters in a human genome. We recognized that we needed to go faster, we needed to be more efficient. We couldn't just use big laboratories filled with instruments the size of refrigerators. We needed to have fancier methods, smaller methods, more efficient methods. And in fact, those are already here now. In instruments that would fit on this podium, we can now sequence a human genome for something like $1,000, and we can accomplish it not in years, but in a matter of a couple of days. We will quickly get this down to a day or so. And in fact, what's remarkable is that the technologies that we use today for sequencing a human genome are not the ones we'll be using 2, 3, 4 years from now. Better and better technologies are coming available all the time. And in fact, they're getting smaller, they're getting simpler, and they're able to be used in settings that don't require significantly expensive laboratories. For example, I hold in my hand a device that can now be used to sequence DNA. In fact, this little device is the entire laboratory that you need to sequence DNA. It just has a little chip inside that does everything you need. Well, the only other thing you need is you actually need a laptop computer, such as the one sitting here, because this is a USB port. You plug it into a laptop and you can sequence DNA with this device. No longer do we need whole laboratories. No longer do we need large machines. And what that means is that, especially for smaller genomes, such as ones that will be discussed at this conference, viruses and bacteria in particular, where this device absolutely works well, we can now imagine sequencing genomes in that setting in places that don't have strong infrastructure. This device takes advantage of a technology called nanopore sequencing. I won't go through the details except to say it reflects some incredibly innovative ideas developed by scientists over the last 15 years. This exact device that I hold in my hand was used in Africa recently to type the strains of Ebola during the Ebola outbreak. All that was needed was this device and a laptop computer. Similarly, this device has been used in Antarctica, hardly a place with strong laboratory infrastructure. The same for the International Space Station. This exact device has sequenced DNA on the International Space Station. And so what that means is we have incredible potential now for using devices like this for sequencing microbial genomes, and eventually with further refinement, we hope devices like this will be useful for sequencing human genomes. And in fact, the same company that puts out this device claims that they will soon have a device that actually attaches to your smartphone and will be able to sequence genomes. We will see when and if this actually comes to pass. But the imagination is sky high with respect to newer and newer approaches for sequencing DNA. While having in hand cheaper ways of sequencing DNA meant that we couldn't just stop at sequencing one genome like the Human Genome Project did, but now go out and sequence many people's genomes. And in fact, we've now sequenced tens of thousands and probably hundreds of thousands of human genomes have been sequenced. We've amassed catalogs of the different spellings that each of our genomes have. Each of us carries something like 3 to 5 million letter differences in our genome compared to, say, the person sitting next to you. And we now have on the internet, widely available to anybody worldwide, data resources that contain information of the most common variants that exist in all of our genomes. We're increasingly able to look at those variants and figure out what they do in changing the function of the genome because we have profoundly advanced our understanding of how the human genome actually functions. We still have a long way to go in completely understanding all of the code in those 3 billion letters. But in the first 15 years of analyzing that code, we've learned about the 20,000 genes that are in the human genome. We've learned a lot about the sequences that determine when, where, and how much those genes get turned on in different cells and in different tissues and at different developmental stages. And increasingly, we're learning how the environment interacts with our genome and makes epigenomic changes, these different ways that DNA can be modified that influences genome function. And so while we certainly have a lot more to learn about how the human genome works, increasingly we are learning more and more and are able to make remarkable inferences about genome function. And what that has provided us is an ability to understand which spelling differences play a role in our health. And we've made significant advances in unraveling the genomic basis of human disease. We've seen this most profoundly in our ability to understand the genomic basis of rare diseases, diseases that are caused by defects in a single gene. I will give you an example. The day the Human Genome Project began, there were 61 rare diseases for which we knew what was broken in the genome, what gene was mutated, 61. Fast forward to today, and that number is 4,800. 4,800 rare diseases, we now know exactly what is wrong in the patients, and that number grows every single week. Similarly, we are using the technologies of genomics and knowledge of the human genome to better understand the genomic basis of diseases like cancer, and increasingly getting clues about more complex diseases that have many different genomic contributions to the disease, as well as greater influence of the environment, diseases like hypertension, diabetes, Alzheimer's, autism, cardiovascular disease, and so forth. So again, we have a long way to go, but in the first 15 years of having the human genome sequence, we've made significant advances in unraveling the genomic basis of disease. These 5 highlights together have set up a circumstance that we are just beginning to see how genomics can be used to change the practice of medicine. We now have vivid examples of genomic medicine in action that are emerging. And let me tell you, that has come faster than most of us ever expected that it would. Once we had the new technologies for sequencing human DNA, it actually turned out that generating a sequence of a patient's genome was relatively straightforward. It was almost trivial. You could get all of the letters lined up of a given patient that you were taking care of. However, understanding that sequence remains still a great challenge. When you round on that patient in the morning, many times this is what you feel like. You don't really know exactly which of the 3 to 5 million spelling differences in their genome is clinically important and which is not. But slowly but surely that gap is closing, and increasingly we are beginning to learn how to interpret that information, and with that, genomic medicine is becoming in focus. We are beginning to see real uses of genomic information as part of clinical care. Let me just briefly tell you about a few examples, what I think are the hottest areas in genomic medicine today. This list will grow with time, but I just want to give you a flavor of what I think are some of the most exciting early examples of genomic medicine in action. The first absolutely relates to cancer. Cancer is a disease of the genome, and the tools of genomics are perfectly tuned to being able to make much better inferences about the specific cancer in a specific patient. We now can open up every single cancer, and we could read out the changes that have taken place in that cancer genome, and we have learned that the insights that are gained from genomic information about individual cancer specimens is immensely valuable for diagnosing the exact type of cancer and really paving a way to more accurate ways of treating individuals with cancer. I can absolutely assure you that genomics will become everyday use in the practice of medicine when it comes to cancer diagnostics and care. Another absolute example that is here and now is the use of genomics to better select medications for individual patients, an area known as pharmacogenomics. Here it is very clear that all too often we just generically prescribe medications to patients even though we know that in many cases that medication won't work, or worse yet, it will make the patient sicker. But we've come to learn that some of specific genomic variants are the ones that influence our response to medications, either making us a good responder or a bad responder. And now we are learning how to gain genomic information in advance of making decisions about which medication to give a patient, and that is proving to be quite effective. Thailand is a model example of this. A disease found in this country, occasionally Stevens-Johnson syndrome, is due to specific genomic variants that predispose individuals to horrifically bad reactions to certain medications, but through a program that has been instituted in this country, Stevens-Johnson syndrome's rate has gone down considerably. They are using pharmacogenomics absolutely in real time. They are a model for the rest of the world. A third hot area of genomic medicine is for the use of rare genetic disease diagnostics. We always envisioned that maybe one day, if you actually had a patient's blueprint in front of you, you could figure out what was wrong with that patient. But we really needed to figure out exactly how to do that, but we have figured that out over the last 10 years, and particularly over the last 6 years, where now patients with undiagnosed conditions, when you simply don't know what is wrong with the patient, you sequence their genomes. Right now, about 30 to 50% of the time, you will figure out exactly what is wrong with them. I think that percentage will go up as we get better at interpreting the human genome. But I can tell you now that worldwide, hundreds and hundreds of patients with rare genetic diseases are having their genomes sequenced as part of routine diagnostic workup every single month. Finally, the last area I would point out that is hot, and in fact, it probably is the #1 use of genomics medicine today, is in the area of prenatal genomic testing. In this case, prenatal testing has been going on around the world for decades, most typically looking for abnormal numbers of chromosomes, such as trisomy 21. But that has involved invasive methods that risk the viability of the pregnancy or are in some cases a little unpleasant, such as amniocentesis. But it turns out that these exquisitely sensitive methods for sequencing DNA that have been developed in the last decade now allow us to detect the very small amounts of fetal DNA that float around cell-free in the maternal bloodstream. So now, through a simple blood draw of a pregnant mom, we can basically get all the same information for screening for these chromosomal anomalies that we previously had to do more invasive procedures to access that fetal DNA. The uptake of this non-invasive prenatal testing has been incredible, and in fact, it is predicted that in 2018, 4 to 6 million pregnant women will have this genomic test done this year alone, and that number is expected to rise in future years. So what I would contend is that these 4 areas, which are just some of the earliest areas of genomic medicine, nicely illustrate that we are well down the road towards being able to realize genomic medicine, starting with the completion of the Human Genome Project. In the last few minutes, though, let me tell you about a yet more exciting opportunity that we think is on the horizon and looking towards the future, is the recognition that diseases are not all about genomics, that diseases are more complicated than that, and we recognize that genomics is one part of this complicated equation, but that environment and lifestyle and social factors play a major role in human health and disease as well. What I've told you about in the journey of genomics is untangling the left side of this bar graph, of this pie chart, and I'm able to talk about it because we've seen this remarkable technology surge that have allowed us to understand the genomic basis of human disease. But inspired by the technological advances in genomics, we can be equally inspired because we're beginning to see better and better technologies for measuring aspects of our physiology and our environment, mobile health devices, and so forth. And we can expect a similar technology surge in getting better and better ways of getting data about our lifestyle, our environment, our physiology, and so forth. This has led to the notion of expanding just beyond genomics to being more precise about everything about disease. The G's, A's, T's, and C's of our genome are critically important, but so are some of these other parts of our everyday life. And so precision medicine is just a little broader than genomic medicine. It is basically just more precise accounting of individual variability. I've told you about the individual variability that we get by studying patients' genomes, but we similarly now are seeing technologies coming available for measuring more precisely people's physiology. They're using little devices that they can wear, their heart rate, their blood pressure, some blood analytes, and so forth. Similarly, better technologies are becoming available for accounting for individual variability in how much we exercise, how much we sleep, what pollution or what chemicals we're exposed to, what we are eating, and what we are doing to our bodies on every day. And one can imagine if we got enough data about all this variability and we studied it, maybe we'd be able to tease out a lot of subtle contributions to human disease. Imagining that, if we were going to implement this, we need to move beyond the way we practice medicine today, a medical care system that is largely based on the expected response of the average patient. But we know none of us are average. We're all a little bit different: genomics, environment, exposures, lifestyle, and so forth. Could we imagine a tomorrow where medical care would be based on individual genomic, environmental, lifestyle differences? All of this would give us a more precise way to prevent and treat disease. But how do we get from today to tomorrow? Many people around the world are interested in this problem. One individual in the United States who was particularly interested in this was our former president, President Barack Obama, who in 2015 announced the launching of a new program in the United States called the Precision Medicine Initiative. It was actually quite analogous to other efforts in other countries. In this particular case, he wanted to jumpstart this in the United States, recognizing the potential of precision medicine that I just described for you. The key component of this initiative in the United States is a brand new program that we are just launching called the All of Us program, which, like the Human Genome Project, is audacious in scope and scale and will involve many, many researchers in the United States and around the world. In the United States, we will be getting 1 million of our citizens to volunteer to participate in this program. These individuals will share genomic data, lifestyle information, they will provide biological samples, and all of this will be a link to their electronic health records. And we will follow these people for many, many years and collect massive amounts of data on them. And then we hope that scientists around the world will analyze this data. We will share this data with them in an open, responsible way. We will be building in strong privacy protections as well. Because we believe there's an unparalleled potential now to use complicated tools of data science to all at once analyze genomic data and electronic health record data, as well as data coming out of new technologies that are going to tell us lots of other aspects of our everyday life. And together in doing so, we will gain greater insights about various components of human disease, which will pave a way for being able to practice medicine more precisely. If you're interested in reading more about this new program, which I think is very analogous to the Human Genome Project, I would point you to this webpage, and there's a lot of informative information you could read about there. So I hope what I have accomplished is to tell you first about why the Human Genome Project is being honored in the field of medicine by this prestigious award, for the foundation that it has laid for what has transpired in genomics over the last 15 years as we head towards an era of genomic medicine. But I hope I've pointed out to you that it will not stop with genomics. There's an exciting new frontier ahead of us that includes information beyond genomics, other information about our lives, that if we can capture that information and understand it, we will one day have much more precise ways to practice medicine. Thank you very much for your attention.